Assalamu alaikum. My name is Huma Hasnain. I'm from 4th Prof Evening. My roll number is EP1770060 and the topic of my presentation is forced swim test, in which I'm going to explain behavioral coding and biological factors. Protocols in FST. Abstract. Forced swim test in rodents is a widely used behavioral test for screening antidepressants in preclinical research. Transitional value of preclinical studies may be improved by appraisal of the quality of experimental design and risk of biases, which remains to be addressed for FST. The present protocol of a systemic review with meta-analysis aims to investigate the quality of the preclinical studies employing FST to identify risk of bias in future publications. In addition, this protocol will help to determine the effect size for primary and secondary outcomes according to the several aspects of the FST study design. The protocol details using the FST in rats, which takes place over 48 hours and is followed by the video analysis of the behavior. The swim test involves the scoring of active swimming and climbing and passive immobility behavior when rodents are forced to swim in a cylinder from which there is no escape. There are two versions that are used, namely the traditional and modified FSTs, which differ in their experimental setup. For both versions, a pre-test of 15 minutes is included. Introduction. Major depression disorder (MDD) in human is characterized by depressed mood and behavioral inhibition and often comes with social avoidance, generalized anxiety, eating disorder, sleeping and problems to cope with stress. Despite the difficulty in finding suitable model to mimic subjective, behavioral and neurobiological aspects of MDD, there are several animal models predictive of MDD treatment. Most of these animal models are based on behavioral responses of an animal to inescapable stress, providing a framework for several laboratory tests. Usually, inescapable stress induces behavioral inhibition or immobility that can be counteracted by antidepressant treatment. Therefore, behavioral tests in animals are employed as screening step during the preclinical phase of antidepressant drug discovery. Forced swim test in rats and mice used in preclinical trials of antidepressant FST is easy to run, inexpensive, sensitive, and relatively selective to run to known antidepressant. Now we have the behavioral coding or observable behavior. For rats, code the 5 minutes of the test stage. Code the duration of time spent as immobile if the mouse is floating with the absence of any movement except for those necessary for keeping the nose above water. Code the duration of time spent as struggling climbing if quickly movements of the forelimbs are observed such that the front paws break the surface of the water. Code the duration of time spent as swimming if movement of forelimbs or hind limbs in a paddling fashion is observed. Note, it is possible to use an alternative way to code these behaviors. This includes a time sampling method. Rate the swimming, struggling or immobility as the frequency of episodes at 5-second interval throughout the test session. Other methods of behavioral coding. 3 commonly used coding methods include momentary time sampling, partial interval recording (PIR), and event coding. Event coding, also called frequency counting, is often viewed as the most precise observational method for assessing the quantity of behavior because the observer records each instance of the behavior. However, these discontinuous methods of observational coding only provide estimates of behaviors because they don't record each behavioral occurrence. MTS involves coding a behavior that only occurs at a specific point in time during a specified time frame. For example, the observational point in time may occur every 10 seconds and only behaviors that occur at the moment in time. A variety of factors contribute to decisions about the selection of behavior observation and recording systems, including the available resources, the complexity of the coding scheme, and the type of behavior observed. Compared to PIR, MTS may be more accurate for observations of the duration of behaviors. Behavior analysis. In our laboratory, we upload the videos files directly from the camera to a PC and do the analysis on the PC. The most important aspect of behavior analysis, and usually the biggest source of variability between observers in the FST, is the correct identification of movements that are counted as bona fide mobility. Our operational definition for mobility in the FST is any movement other than those necessary to balance the body and keep the head above the water. Mice generally float in water readily. However, they still manifest small movements to balance their bodies and keep their heads above the water. These behaviors are not an attempt to escape and shouldn't be scored as mobility. Also, after a single bout of mobility, even though essentially immobile, mice can still drift in the water as a result of a momentum. These movements also shouldn't be scored as mobility. An inter-observer reliability test should be conducted for every new observer before beginning to collect the data from test animals. In our laboratory, each new observer first watches an experienced observer scoring. After the new observer gains confidence to differentiate mobility from immobility, They then score with the experienced observer watching the pointing out any mistake. Once this phase is successfully completed, the new observer will analyze a specific set of FST videos that we keep in our laboratory for training purpose. Only after a high level of inter-observer correlation is obtained with the experienced observer does an investigator start analyzing FST videos in actual experiments. We archive the data from these training analyses to constitute internal standard for the laboratory for future use. We have observed differences between strains in the manner in which they express mobility and immobility behaviors and mean immobility time between sexes. When a new strain, sex, or genetically modified mouse model is tested in the laboratory, it is necessary to again undertake this type of reliability analysis. Biological factors. Many biological factors affect behavioral performance during the FST and might 13-14 months away. Age. Evidence suggested that there was a strong difference between the younger and older mice groups in the FST. Older 8 to 12 months old, naive male, this is the C57BL and /6J, this is the genome. Mice had lower immobility than their younger 2 to 3 month old counterpart. Sensitive to some antidepressants is also profoundly altered by the age of mice. Tricyclics, noradrenaline reuptake inhibitors, and serotonin reuptake inhibitors exhibited stronger effects in 4-week-old Swiss mice than 40-week-old mice. Social isolation as well as treatment with reserpine and antihypertensive and antipsychotic drug increased FST immobility in 17 to 21-day Swiss Webster mice, but not in 26 to 30-day-old mice. Mice susceptibility in swimming. It was recently found that the mice susceptibility in swimming proficiency is another important factor influencing the mice FST. It has been reported that male mice that are genetically selected for long attack latency LAL and short attack latency SAL display differences in the structural and functional properties of postsynaptic serotonergic 1A 5-HT1A receptor that they also show divergent behavioral responses during the FST, such as higher immobility in LAL and lower immobility in SAL mice. Here is references. That's all from my side. I hope you like this presentation. Thank you. Allah Hafiz.